Okay, it's a few minutes past 7, so good evening and welcome. Thanks very much for joining us for tonight's Ask the Expert event, which is being hosted by the National Institute for Health Research, NIHR, the nation's largest funder of healthcare research. My name is Peaches Golding, and I'm the Lord Lieutenant for the City and County of Bristol. That's the Queen's personal representative here in the county. And I will be chairing tonight's event. Someone asked a question, and guess what? Shirley, I'm currently taking part in the Comm vaccine study. It's the first one looking at the effect of using a different vaccine for the second dose from the first dose, in addition to 2 different intervals between doses. So although we have approved vaccines, vaccine research is still really important. During the next hour, we aim to share what is known about COVID-19 vaccines and the role research can continue to play, as well as answering any questions you might have about COVID-19 vaccine research. So tonight, let me introduce to you our panel of experts. First up, Professor Saul Forst is Professor of Pediatric Immunology and Infectious Diseases at the University of Southampton, Director of the National Institute for Health Research Southampton Clinical Research Facility, Clinical Director of the Clinical Research Network, Wessex, and an NIHR Senior Investigator. We have Professor Adam Finn, who has worked on delivering a number of vaccine trials, including the Oxford AstraZeneca, Janssen, and Valneva. And he also sits on the Committee of Vaccination and Immunization. Next up, we have Dr. Lawrence Barnes from Rame Group Practice in Cornwall. He is the he is the local principal investigator for the PROVENT study, which is looking at whether giving antibodies to people can provide protection for those who are not eligible for the vaccine. And we have Lisa Berry, who is a research matron in the NIHR Southampton Clinical Research Facility. As the acting clinical nursing lead for phase 1 accreditation, Lisa's responsible for ensuring that the clinical and research care delivered is of high quality. Lisa is a member of the UK Clinical Research Facilities Network Intensity Working Group, a committee chair for the International Association of Clinical Research Nurses, and is a founding member of the UK branch of this association. Now, we've already received some great questions. If you have any further questions, can you put them in the Q&A box at the bottom of your screen, and we'll try to answer just as many as we can do. Our panel tonight, as you've just heard, are experts in vaccine research, so we won't be able to answer questions on other topics such as policy or rollout. We've also had a number of questions from those in the Novavax study, but as we only have an hour and lots of questions to get through, the NIHR team will share an email address in the chat box below so you can get in touch with them directly and they can answer your questions personally. So before we move on to the Q&A section, let's hear further from Professor Adam Finn first and Professor Saul Forst, who will be telling us about where we are with COVID-19 vaccines.
Okay, well, thank you, Adam. Now, I'm going to have to refer back to her talk. Instead of referring back to Adam at times, I'll refer forwards to what Adam's going to tell you instead. But I think that'll work and it should all be fine. So I'm going to explain what we're doing at the moment nationally for COVID vaccine research and the trials that are ongoing and the ones that we are going to be doing over the next few months and why. So Adam will explain in a little while exactly how the COVID vaccines work, but you'll know from the news that the Vaccine Task Force in the UK has secured a number of different vaccines for use in the UK population, many of which we've been conducting trials for here in the UK. And so far, we have three vaccines, the Moderna vaccine, the AstraZeneca vaccine, and the Pfizer vaccine, that have been approved for deployment under the Regulation 174. That's not a full license, that's an emergency deployment. And Janssen has now been approved on a one-dose schedule in the US, EU, and by the World Health Organization. And we're expecting Novavax to receive emergency authorization as well, some point in the next week or two, we hope. Now, all the vaccines prevent hospitalization and death, but so far, the AstraZeneca-Oxford vaccine is the only one to demonstrate some effect on the carriage, the asymptomatic carriage of transmission, although data is emerging for that from the Pfizer vaccine, where it's just been used as a single vaccine in Israel, and will become clear as we move through the pandemic. It's really important to realize that we can't compare the trial data directly, because it's a bit like comparing apples and oranges. The different studies use different definitions of COVID. And so, and they work in slightly different ways. So those lower headlines from the trials really don't seem to matter much. And Adam's going to come back to some of the real world data that shows us that all of the vaccines that are being approved look to be pretty good at stopping COVID disease, particularly hospitalization and death. And you know that for the vaccine deployment agreed by the JCVI was that we were going to focus in the UK on the priority groups of whom 99% of the deaths of COVID are from, and the JCVI made a decision based on the science available at the time to go to 12 weeks for the second dose in order to get as many people as possible to have their first dose to try to reduce the impact on the NHS by preventing hospital admission and death. And again, Adam's going to come back to that in his talk in a bit. So what do we need to do now? What are the ongoing questions? Well, we've still got some vaccine task force supported phase 3 trials. We've got trials of new vaccines, and we've also got an ongoing need to look after all our trial participants who've participated so far. And as the vaccines have been deployed, as people are invited to receive their NHS vaccine, people have had the ability to unblind themselves and decide if they're in a placebo group, the dummy trial group, to take an NHS vaccine, or they'll know that they're protected from their trial vaccine. But for the people who haven't yet received their invitation to have NHS vaccine, those studies, so the Novavax trial and the Janssen trial, that are still that still haven't been approved yet for deployment. As they are approved for deployment, there'll be a crossover for people in the current trials to be able to, for people in the placebo group, will receive a dose of the trial vaccine that will then protect them. So, for example, if you're in Novavax placebo, you'll be offered to take the Novavax vaccine, and if you're in Janssen placebo, rather than the 2-dose study, you'll be offered 1 dose of Janssen because that will be the approved dose. Now, what that means is that the trial integrities will be able to be maintained, and the companies and the investigators will be able to still look at all the safety data, all the immunology data from those vaccines. We just won't have the control groups because in a pandemic, it wouldn't be ethical to leave people without vaccine at all. So the crossover design is a way of keeping people within the studies and actually looking at the integrity of the vaccines themselves and their immunology and their side effects. We need to look after people if, and it's a big if at the moment, there's a vaccine passport scheme. We need to make sure all our trial participants are not at a disadvantage, and that's work with the NHS to make sure that happens. And of course, we've got ongoing safety and immunology. So then we've got new studies done by the National Vaccine Immunisation Evaluation Consortium, which are going to tell us what the NHS needs to know for the autumn. And I'm going to briefly tell you about that. Trials in pregnancy, trials in children, trials in immunosuppressed people with different conditions, and human challenge studies. And I'm very briefly going to tell you about each of those. So the current phase 3 trials are Janssen, which is nearly finished for the 1 versus 2 dose study. The Valneva phase 3 study will be a study of that vaccine compared to the Oxford vaccine. I don't know whether Adam's going to talk about Valneva, but that's a whole virus killed vaccine that's killed. And it might be that Valneva can protect against mutation strains as well as the current strains of the virus. And then the Medicago vaccine is another one in a similar technology to the Novavax, which will be following on in April. So the first of the NICEX studies is recruited already. This is the Comm study led by our colleagues in Oxford, Matthew Snape. And this is recruiting people aged 50 and over, and focusing on people also with mild to moderate comorbidities and conditions, and a diverse population representative of the UK. And this study is to study the schedule. So give the first dose of Oxford or Pfizer, and then the second dose of both those vaccines, but also the other vaccines, and compare the schedules at 4 and 12 weeks to inform how the deployment can carry on, but also help inform the rest of the world. So the idea is that one group get Oxford and then Oxford with 4 weeks or 12 weeks, another group get Oxford and Pfizer, both 4 weeks and 12 weeks, and then the same for Pfizer, and Pfizer Oxford. And then there'll be CommCov2 later on in the summer. As other vaccines become deployed, CommCov2 will then look at the new vaccines that receive their MHRA approval. Adam and his team, Rajeka Lazarus, is going to lead this study in Bristol, the Comm Flu Cov study. And this study is going to provide data to inform whether we can give the vaccines, the COVID vaccine at the same time as an influenza vaccine. And this is going to inform both NHS delivery and workforce planning for the autumn. We don't mean mix the vaccines and give them as one injection. We mean be able to invite people to one visit and give one vaccine in one arm, and if it's COVID vaccine in one arm and flu vaccine in the other. And this is quite complicated because we have different flu vaccines used in those people under 65 and those people over 65. And so both of those groups will be included, and people will be given the chance to, if they've already had one dose of Pfizer or Oxford, balance between the two groups to come and receive the second dose of their COVID vaccine alongside a flu vaccine. And that's going to be looking at the side effects and immune function of doing that, and hopefully will show that it's safe to give flu vaccines at the same visit as COVID vaccines. And we know that, we think we know that's true for Novavax. They haven't released their data yet, but Novavax did that within their main study. So we don't have to do that again for that vaccine if it's approved. And then the third study is the Cockpit study, which will be leading from Southampton. And this is going to be in the summer, in June, July, and August, where we're going to look at giving a third dose of COVID vaccine to people who had 2 doses of either the AstraZeneca or Pfizer, there's a typo on the slides, COVID vaccines in December or January. So we're going to be looking at a third dose of the current vaccines, new vaccines against the original strain virus, so the ones that aren't yet deployed in the NHS, and then hopefully later in the summer, new vaccines against the mutation strain of virus. And we'll be looking at 2 groups, the elderly over 75 and healthcare workers, again, with the objectives of looking at the side effects as people are given a third dose, and the immune function, looking at their antibodies. And these 3 studies will provide data for the JCVI and the chief medical officers to decide whether or how to give COVID vaccines to people in the autumn if it's decided that we all need a booster, or some people need a booster. So pregnancy studies are going to start up in the UK soon. The Janssen and Pfizer and AstraZeneca studies will be getting going to provide that data and safety and immunology data. And we've already started the AstraZeneca, Oxford and Janssen studies for teenagers and soon to be younger children. And the question for children are what dose and what are the side effects? Because these are quite immunogenic. They're quite reactogenic vaccines. They cause quite a lot of sore arms and fever. So we've got to get the dose right before we deploy widely to children. And we've got to check the side effect profile in proper trials. And in the future, we might get to ask the question, do the vaccines reduce transmission in the real world, looking at children and schools? But that all depends on what happens in the pandemic over the next year. Immunosuppressed people, there's been a study of the monoclonal antibodies, the PROVENT study as a preventer and the storm chaser as a, what we call post-exposure prophylaxis. These are AstraZeneca studies that PROVENT's now closed, storm chaser's about to close. These might be good products to use instead of vaccines in the immunosuppressed population. And the Octave trials, a national study being led for Birmingham, looking at the routine schedule of people being offered, people with the 4 conditions on the slide there, who have been offered NHS vaccine and studying their immunology in their routine immunizations. Finally, the live viral challenge trials can potentially accelerate vaccine development. There's extensive UK experience for doing this with bacteria and parasites where there's an effective treatment. COVID's a bit different because we don't have an effective treatment. So it's very important that we've done detailed public work, public involvement work to demonstrate the feasibility and the public acceptance of such studies. The UK collaboration involves multiple institutions, including Imperial College London, but also Bristol and Southampton investigators. And these studies will involve young, healthy participants with no COVID risk factors to minimize the risk. The first thing is to develop the model and show that we can do this safely in an inpatient environment, and then the new vaccines might be able to be tested within that system. You give people the vaccine and then challenge them with a standard dose that's been worked out and work out whether the vaccines are effective or not before you go in and do a very large, expensive and time-consuming larger trials in humans. So in summary, the key issues we need to know for the NHS for the autumn are, will a third dose booster be required? And we're going to generate the data for antibody immunity and T-cell immunity. We need to know which vaccines are going to be able to be used if we do want to do a third dose. And we need to know about flu and provide information about different age groups. And so that the JCVI can make a decision about whether to immunize just some or all people. And in the future, we'll hopefully understand more about vaccines in children. And it's possible that JCVI will decide at some point later this year or next year to deploy vaccine in children to potentially stop transmission in order to prevent virus mutations. But children aren't seriously at risk of death from COVID, and so these are not the priority. Children are not the priority at the moment. So we've got an ongoing need for clinical trials of different vaccines and technologies to ensure the global supply. We need to optimize protection, looking at the combinations, different ages and clinical conditions, address the issues caused by virus mutations and understand what to do with vaccines. Make sure the global manufacturing supply chain produces lots of different vaccines. And we heard today about vaccine supply problems. That's why we need lots of different vaccines from different companies. And somehow we've got to avoid vaccine nationalism. I'm sure Adam will talk to this in a bit too, but it's been heartbreaking that the politics has got in the way of sensible decisions about how we use and share vaccines globally. Adam, I hope you're ready now with your slide sharing, but that's what I was going to say.
Okay, thanks, Saul. I'm sorry to have done this back to front. So I'm just going to give a short overview of the different vaccine platforms that we've got and a little bit of information about how well they work. So my name's Adam Finn, at Adam H. Finn's my Twitter handle. If you're a Twitter person, I tweet about vaccines, please follow. Next slide, please. So I am a member of the JCVI. I also work for the WHO for their European regional working group on the COVID vaccines. And as you heard earlier, I'm involved in a number of the trials of the different vaccines being done that Saul just nicely summarized. Next slide, please. So we've gone from no vaccines at all last November to really quite an impressively long list of vaccines that are either licensed and in use or very close to that at the present time in different parts of the world. We're using the first two on that list in this country. We also have orders in for the Moderna vaccine, which has been approved in the UK. The Janssen vaccine, one dose is now coming through, just approved this week by WHO. And we will be hearing, I think, very shortly about Novavax, which has also produced very promising results in a study led by Paul Heath here in the UK. There are published phase 3 data on the Russian Sputnik V vaccine. There are 2 Chinese vaccines, one whole virion vaccine and one adenoviral vector vaccine that are also showing data to the WHO at the present time. And there's a vaccine that's been produced in India, which is also in use in that country already, produced by Biosciences. Next slide, please. The virus has around its surface these sort of flower-like molecules that you can see at the bottom of the slide called the spike protein. And the vaccines we're using at the moment, with the one exception of one of these 2 Chinese vaccines, really either encode for or consist of this spike protein. So they're inducing both B-cell responses, antibody responses, and T-cell responses to that one protein. The protein binds to the angiotensin converting enzyme 2, which is on the surface of human cells, and that's how it gets in. And there's a particular part of the tip of this trimeric molecule called a receptor binding domain that is responsible for that interaction. And we think that antibodies to that particular part of the spike are really important in terms of protection. Next slide, please. So these are the different types of vaccines that we're now seeing in use that, in order at which they appeared. The messenger ribonucleic acid or mRNA vaccines like Pfizer and Moderna, the adenoviral vector vaccines like the Oxford vaccine, Janssen, and the Russian vaccine. Then recombinant proteins, where you're actually giving the protein. I'll explain what these all means in a minute. And that's like the Novavax vaccine. And then finally, good old-fashioned whole inactivated virus vaccines like this Chinese vaccine and this Valneva vaccine that Saul mentioned that we're trialing in the UK. There are others in the pipeline, but they're not yet on the scene. So they're in small font at the bottom. Next slide, please. So wholesale vaccines, we've been using those for more than half a century now. The famous Salk inactivated polio vaccine, or IPV, is the first example of this. But actually, most of the hepatitis A vaccines we used are also this kind of been around for 20 years or so now. Wholesale vaccines, so they're produced by literally culturing the virus, then inactivating it so that they can't cause an infection, but the whole of the virus is there. It's picked up by the immune system and results in immune responses, often with an adjuvant to stimulate those responses. Next slide, please. So this is a tried and tested technology, but it does need to be done right. There are examples of these vaccines that don't work so well, and we are always concerned to get the right kind of immune response. They're cheap to manufacture, and there are plenty of places set up to do that around the world. They contain all the antigens in the virus, and that's theoretically advantageous in giving you a broader immune response. And they're usually easy to handle and transport, so plenty of advantages, but they take longer to develop, which is why we're only just in phase 1 with the Valneva vaccine, while the other vaccines that we're using are already out in the clinic. We do need to avoid antibody-enhanced disease and Th2 type responses, so we need to be careful that we induce the right kind of immune response and don't have a repeat of some of the very early vaccines of this kind, which caused enhanced disease when they were given. And so a lot of care taken to avoid that problem. Next slide, please. For the other kinds of vaccine, you need just to be reminded of the steps between having DNA and RNA and protein in your cells. So the DNA is in your nucleus of your cells and codes for all the genes in the organism. You transcribe that into messenger RNA in your cytoplasm, and that is then translated into proteins. And so you can make a vaccine nowadays in any of these three. Next slide, or next forward. Yeah, so adenoviral vectors are giving you the DNA, which codes for the S protein to your cells. The RNA vaccines giving the RNA, and then the recombinant proteins and whole virus vaccines actually providing the antigens in the way that more conventional vaccines do. Next slide. So hepatitis B is an example of a vaccine of a recombinant protein, as well as the human papillomavirus cervical cancer vaccines. You basically take the gene of the protein of the virus and you grow it in another cell in a bioreactor, produce large quantities of that, and then package it up with an adjuvant and make it into a vaccine. And the Novavax vaccine, and in fact a more slowly coming forward candidate from Sanofi Pasteur, are using this technology. Next slide, please. So this, again, is a tried and tested technology. Lots of plants around the world capable of doing this, relatively cheap to manufacture. However, these vaccines may not be immunogenic, and you need the adjuvant oftentimes to get a good response. Once again, they're easy to handle and transport, so usually at refrigerator temperature. Next slide. What about the adenoviral vectors? So this is a much more complicated way of doing things, a very new technology that we're seeing these vaccines in widespread use for the first time. You essentially use a virus to smuggle the DNA for the virus you're interested in into the cell. But first of all, you paralyze that virus so it can't itself cause an infection. And we use these adenoviruses, which are DNA viruses. You take out some of its genome so it can't reproduce, but it can still get into the cell. And you put the DNA for the spike protein into that vector, and then you inject it and it gets taken up by the cell, and then you make the S protein, which your immune response is then generated against. Next slide. This is another picture showing an adenoviral vector going into a cell, going into the nucleus, then being transcribed into RNA and then making protein. Next slide. So these vaccines came through really fast. They could simply get the code for the virus and manufacture the virus. The information that they needed was simply a code that could be sent in a text message or an email. They didn't need the virus to do this. And these also can be reformulated, as we may well need to do now with new variants, relatively quickly. Turns out these can be relatively easily stored. When Saul and I were first doing these studies last year, we were storing these vaccines at very low temperature, but they can in fact survive quite nicely in a fridge. Now, it is a new technology, and new technologies may have unforeseen downsides and problems, and we may well get into some Q&As about this scare this week around possible side effects, although in fact none of us think there is a real problem with side effects at the moment. But in principle, any new technology could turn up problems that we don't expect. And then finally, the adenovirus vectors may induce immune responses, and that may also cause problems with using further doses of the vaccine. If you've made immunity to the vector, it might stop those boosters from working. Hence the usefulness of these kind of mix and match studies that Saul told you about. Next slide, please. Finally, the mRNA vaccines from Pfizer and Moderna. You're just giving mRNA. It's a very fragile molecule, difficult to make into a vaccine, but turns out to be highly effective. Next slide. So this gets into the cell, but only if you can put it into a lipid nanoparticle, which is a little bubble of a fatty-like material that protects the RNA inside and smuggles it into the cell. The RNA on its own would be too rapidly broken down and wouldn't work. So it's this lipid technology that's enabled these vaccines to be used. Next slide. So these also were developed extremely fast as soon as they had the genetic code of the virus, and likewise can be reformulated, and work is going on related to that at the moment as we watch anxiously for these new variants coming through. And we have certainly seen that these vaccines are highly effective, at least in the short term, and we're gathering evidence on longer term protection. Once again, it's a new technology, so theoretically problems could show up that we don't know about, and we have to look out carefully for that. And as I mentioned, it's a very unstable molecule, and we're having to store these vaccines at very low temperatures, which creates challenges, costs, and of course, particularly challenges for using them in low resource environments. Next slide. So this is just an example of the kind of endpoint of one of the protocols. And you can see that what we're looking for is whether the vaccines prevent disease. On the left, you can see sort of respiratory symptoms that people had to have, along with a positive PCR to count as a case. And on the right, you can see more mild and general symptoms, a combination of which in this particular protocol was enough. So someone with headache or muscle pain and a positive PCR would qualify as a case. So these efficacy measurements initially were really about relatively mild disease in most cases in relatively young people. Whereas of course, what we really need to know about is how well these vaccines protect old people against dying or getting very seriously ill. Next slide, please. And so these are the residual questions, and we have at least got answers to the fourth of these, which I'll show you in a moment. But we need to have a good blood test that we can use to predict whether protection is there, and we're working on that now. We need to know how long the protection lasts after these different numbers of doses and what the optimal dose interval is. Our best guess at the moment is 12 weeks in order to maximize both short and long-term protection. We need to know that these vaccines protect the most vulnerable people who are most likely to get sick. We need to know whether or not they're safe and effective in children and pregnant women, and we need to know about these mixed schedules. And not on this list, we really do need to know how well they interrupt transmission at the population level. Next slide. Very quickly, some data from Bristol. We've been looking over the last 3 months at the admissions with pneumonia to our hospitals, and we've done what's called a case control study. What this slide shows you is that the people we were looking at over the age of 80 were people who were very much vulnerable. Many of them care home residents with high scores in comorbidities and high levels of frailty. So these were old and vulnerable people. Next slide. We found that the Pfizer vaccine was 70% effective in this group at preventing hospitalization after just one dose, looking at 14 days after that first dose. Actually slightly more than that in a corrected analysis of 71%. Next slide. For the Oxford vaccine, the figures were slightly higher, 77%, adjusted up to 80%. So really impressive protection against hospitalization. And then next slide. And finally, when we compared the same period of time from January for the Pfizer vaccine, excluding some of the biases that were present in the December cohort, this also came out at almost exactly the same at 80%. So we're seeing very high levels of protection in the most vulnerable people. Next slide. So we've had the first licensed vaccines less than a year after the virus was first reported. That's a completely unprecedented pace of progress. But we do have substantial challenges ahead in delivering and communicating around these vaccines to get the whole human population protected worldwide as soon as we can. Thank you. That's the end.
Thank you so very much, both of our speakers. You've given us some really helpful information and background, some of it extremely technical according to some of our participants, but all of it extremely interesting. Now, let's go to some of the questions that have been raised by you. Let me encourage you, if you do have a question, to put it in the Q&A box. So let's start now. Saul, tell us, how were these vaccines produced so rapidly?
So basically by the world doing nothing else other than trying to find and make and test COVID vaccines. So the people who were working on vaccines, people like Sarah Gilbert at Oxford and the companies, put all their efforts at the beginning of the pandemic when the virus was sequenced, that's the molecular signature of the virus was published by the Chinese, into refocusing what they were doing already into making coronavirus vaccines. And then companies started making the vaccines for clinical trials, and the whole trial infrastructure, not just the National Institute for Health Research in the UK, but the manufacturing, the regulatory side, the MHRA, put their efforts to agreeing the protocols, designing the trials, and we were able to start them, as Adam was saying, in Oxford way back in April last year, and Bristol and Southampton got involved very early on in those trials. So there weren't any corners cut. It's just that all the other people and trials for cancer and rheumatology and all the other conditions just got put to one side. And the regulators and researchers concentrated on finding the vaccines and treatments for COVID at a detriment to everybody else, as we now know. But we've got vaccines to get us out. And of course, the studies have been massive. We don't normally have new vaccines tested in 30, 50,000 people. So not only were the studies done sort of in parallel, but the huge numbers for the initial safety data, and the regulators were looking at doing something called a rolling review. So they were looking at all the data in real time. And normally you finish a study, you collect your data, you clean it up, and then you submit it. It takes years to do that, but everything was being done in real time. And in fact, we've probably done the trials bigger and in more detail than other vaccines that have come to market.
Okay, well, that leads us very neatly on to the next question I have here. I mentioned that I'm taking part in the Comm trial. So, Lisa, tell us, we know they play a huge part in what's going on now, helping us to get out of this problem. So how is it that people can take part in a trial?
So people can sign up to take part in trials in a variety of ways. We have the National COVID Vaccine Database, which we've had the details of that and the website circulated in the chat function. So for those of you that are listening and dialing in this evening, if you're interested in taking part in research or want to find out more, please do have a look at that website and sign up. Alternatively, the local site. So for us at Southampton, we're running a large portfolio of these studies. People can sign up through the national database and be contacted via that. But also we will have local communications and material going out via newspapers, websites, social media, posters, and people can contact the team directly to take part. Once you've registered your interest or contacted a member of the team, there'll be usually an online pre-screening process. And this is to make sure that we're not wasting anyone's time. If for example, currently, if I were pregnant, I would not be able to take part in the studies that we've run so far. So it will be a screening process. So if it says, are you pregnant? Or if you're under 18, then no, at this stage, you're not eligible to take part. So at each part of the study, there are a different number of safeguards built into it. So only the right people who are eligible can then proceed to that next step. Once you've got through that pre-screening questionnaire online, you'll have a telephone call with a member of the team who is experienced, knows the study inside and out. We would then invite you into a screening visit. The screening visit takes place in a local location, staffed by our expert research doctors and nurses, where you will have find out all about the study. You'd be consented. We would do a full sort of health check on you to ensure that this is the right study for you to take part. Depending on the study itself, you may have a screening visit and then come back a few days later to have your vaccine, or we can vaccinate you at that visit. The first visit can take about 2 hours. If we're doing a combined screening and vaccine, it can be between 2.5 to 3 hours, depending on how long we're going to monitor you for after that visit. Once you've been vaccinated, we will usually you'll be sent home with a diary to complete for a few days so we can see how you're feeling. Is your arm feeling a bit sore? You've got a 24-hour contact number to contact the team and get through to a study doctor. Depending on the study, we will then follow you up for a period of time. And what I'd like to emphasize is that if you've been unblinded, if you're taking part in the study and you've been unblinded, please speak with your study team and stay in the study if you can. It's really important for us that we can continue to follow you up and see, we're looking at your health assessments, we're looking at your immune system and how long you're having that immune response. So you could be followed up between 1 year to 2 years. Once you get past your initial first and potentially second vaccine visits, your follow-up visits should be very short, take about 20 minutes each time.
Well, thank you very much. I recognize quite a lot of that because I feel very safe having been screened and taken part. I've had my first dose. I have my second dose on Saturday. So we'll see. It's terribly exciting. So now on to the next exciting bit, because there has been a lot of concern about blood clots and the AstraZeneca jab. So what are these based on? Is it a limited concern, or is it something that could happen with other vector-based vaccines? Adam, I think you're probably best to answer this one.
Yeah, sure. So I think it's important to emphasize immediately that there is no evidence at the moment that any of these vaccines causes blood clots. What the situation is that a number of individuals have been described who have developed blood clots after having received actually not just AstraZeneca, but other vaccines as well. And the question has been raised as to whether that might be causally linked. The truth is, though, that illnesses occur all the time, and they don't stop happening when you receive vaccines. If you received your vaccine and walked out in the street and were knocked down by a bus, no one would complain and suggest that the bus was killing you because of the vaccine. But when it's another illness, it raises that question. And so for that reason, these cases are being reviewed to make sure that they are not in some way causally related. But the UK data, and this is the country in Europe that has used far more doses of the AstraZeneca vaccine than any other country, is showing no increase in the number of cases of people with thrombosis of any kind as compared to what we normally see. In fact, the numbers of cases that we have seen after the 2 vaccines we're using are more or less exactly the same and not in any way raised. So we, at this point in this country, do not think that there is any cause for concern about either of these 2 vaccines with regard to thrombosis or indeed any other serious long-term side effects. The only concern that we have had is that a very small number of people have had serious allergic reactions very shortly after receiving, particularly the Pfizer vaccine. But all of those people have been rapidly and effectively treated by the trained staff who are present in the clinics and have made good recovery. We are waiting tomorrow for the verdict of the European Medicines Agency. We're all optimistic and in fact, I would say confident that their conclusion will be, continue to be, that the benefits of the AstraZeneca vaccine are very clear and that using the vaccine is important and it should continue in the context of the pandemic. And I think they will also conclude that there are no concerns about side effects, or at the very worst, that they wish to continue to investigate and to observe and see what's going on. But we'll see tomorrow what they say. For now, the message in the United Kingdom is loud and clear that this is a safe and highly effective vaccine, as I showed earlier, and that it is very important that people come forward and receive their dose as soon as they're invited.
Okay, that's extremely reassuring, both about the blood clots and the allergic reactions that you mentioned. So we'll see what is said tomorrow, and I bet you're right somehow. Let's go to the next question. This one says, my local GP is running a vaccine study. Is it safe to take part in research outside of a hospital? Lawrence, can I direct this one to you, please?
Absolutely. I think if you're lucky enough to be registered at a practice that is research active, then the potential to engage in research in something like immunization with your family doctors who know you, they have your computerized records, they know the medicines that you're taking, and they know all about your past medical history. They are close to where you live. So there's a lot of advantage to that side of things. There's a lot of evidence that practices that engage in research also provide better care to their patients. So I think from our perspective, we think it offers something additional to our group. We have no questions about safety because clinical trials are run in such an organized way that all of our staff are trained both in all of the usual things that you would expect, like resuscitation, but also in trial-specific procedures. So I think absolutely we'd encourage you. One of the barriers sometimes, especially when you're in Cornwall, is going to that hospital a few miles up the road or the other side of a river in our case. So actually being able to take part at your local GP practice is a really positive thing.
Oh, that's wonderful. Very good news. So this question, Saul, I'm going to ask you this one. Now, some people have been hesitant about taking this vaccine because they don't quite trust it. So what's exactly in there? Is there any justification for these fears? I mean, is the vaccine safe for people of, well, adults, because you mentioned earlier children is a different case, but is it okay for adults, people with disabilities, people of different ethnicities? So how confident were the MHRA that the vaccines were safe before they were granted approval?
Fully confident. Peaches. And the MHRA used to run the European Medicines Agency. All the experts in Europe were used to be the MHRA and colleagues and who led the work. And so we know they are globally, if not the best, one of the best regulatory agencies. And we can be fully confident that nobody in the MHRA would have approved a vaccine for use in the UK population without absolutely solid safety data from the clinical trials. Not only that, and as I said earlier, the trials are huge and have been tested in some of the biggest vaccine trials ever done in the world. But since we've deployed the vaccine, the MHRA also have what we call pharmacovigilance systems and monitoring so that doctors and the public report all the side effects in real time on the yellow card scheme, which is now online. And the MHRA make this data available weekly. We can see it, you can look it up. And so all of these databases are available. The MHRA have people looking at them. And as Adam was just explaining for the clots, we can be completely confident that these vaccines are safe in the UK population and that they wouldn't have been deployed unless they were to do so. Now, you asked me a different question, actually. You asked me, are people right to be anxious? I personally, you know, it's a bit like MMR in children. There's so much noise on social media. There's so much false information that it's sometimes hard for people to decide what's true and what's false, which is why we run events like this, which is why Adam spends a lot of time on the radio. I try and avoid it, but sometimes get dragged in. But, you know, we just have to, people who know about vaccines and understand it need to be here to answer people's questions, reassure people and tell the truth. And hopefully people spreading the really malicious false information on social media will become outcasts, because by making somebody anxious and delaying them having a vaccine puts them and their families and the rest of us at risk from catching COVID and transmitting it. So I really, really hope people are reassured by the work we're doing, by this sort of seminar, and people tell their friends to stop spreading nonsense.
Well, that's good. I'm really pleased to hear about your exceedingly high confidence levels and that you continue to take part in these discussions, because it's very important for people to understand at all different sorts of levels of scientific and medical competences exactly what's going on. So
it's easy to see how people can be confused, Peaches, and somehow we've got to work with people. We don't want to be instructive in the UK, do we? We don't want to make it compulsory. That would probably backfire on us.
Mm. Yes, it could very well. Now, Lisa, this is a question that I have been dying to ask, because I'm curious to know, what sorts of responses have you received from people like me that are taking part in these trials? Us volunteers, us patients, as it were. Give us the lowdown.
I'm glad you asked me that question. I'm incredibly privileged to have the role that I have in research and in nursing, being able to work in partnership with people who are volunteering to take part in research, be they healthy volunteers or have long-term conditions, is absolutely a privilege and an honour. Working in partnership with them, we can't progress healthcare. We couldn't have done the research that we've done so far into getting the vaccines licensed and take that research forward for the benefit of the population without people like yourself taking part in research. The response we've had from the public has been absolutely phenomenal. And I feel I've seen absolutely the very best of humanity throughout this pandemic by the response. We have a patient engagement team. We do a lot of work with our participants to get feedback from them, to see what has the experience been like for them. And consistently, we are 99.8% of our participants saying they've had a very good or excellent experience. There are some things we can't control, like the weather or traffic coming in, but generally the feedback from, or consistently always the feedback from our participants is that they've had a great experience, they felt valued, they felt like they've done something, they've contributed to this, the fight against this global pandemic, and they walk away having had a really positive experience.
Well, I'll say I've had a very positive experience so far as well, even though I feel like I've given gallons of blood. But again, to test the immunology and all of those things, that's extremely important, and it's not painful in the least. Okay, I think we are running close to time, but I've got one more question that I'd really like to ask you, Lawrence, and that's around the need for a negative COVID-19 test before receiving the COVID-19 vaccine. What can you tell us about that?
I think that's just one of those myths, really, Peaches, to be honest. The only thing that should restrict you from having your COVID vaccine is either that you are seriously unwell. Even being mildly unwell is not a reason to not be able to have your COVID vaccine. If you are seriously unwell with a high fever and very unwell, or showing COVID signs, then obviously you should not be going out at that point. You should be following all the usual government restrictions and getting a swab done. But if you basically are pretty much well in yourself, or with a very mild cold or something of that sort, you can carry on. The priority is to get as many people immunized as possible.
Okay, that's wonderful. That's very good advice. And you answered that so rapidly. I can ask you let me ask you another follow-on question, which is about advice for women that might be pregnant or breastfeeding. Now, one of our professors gave us a little bit of information about that earlier, but if you could just say something, I'd appreciate it.
As a snapshot, I mean, there are two separate parts to that. The breastfeeding part is pretty clear. We're advising that women who are breastfeeding are perfectly safe to go ahead and have the immunization. We don't have all the evidence when it comes to pregnancy as yet. So it's a slightly more tricky thing to say that everybody should go ahead and have the immunization currently. However, we are saying, and we have colleagues, I have colleagues here, pregnant doctors in our surgery, who because of their exposure risk have decided that the benefits of having the immunization outweigh the risks that they feel are very low of actually having it. So whether it's because of increased health or increased exposure risk, it becomes an individual choice. But at the moment, it's not an overarching thing as it is for breastfeeding.
Okay, wonderful. Thank you very much for that reassurance. We do have time for another question. I'm going to run this very, very close to the wire, you can tell. But Saul, do you experience worse side effects from the vaccine if you've previously had COVID or not? And how do the vaccines impact on people that have long COVID? We hear there are quite a lot of people that are suffering from this long COVID.
Yeah, so I think at the moment, the recommendation is that everybody, as Lawrence has just been saying, everybody should receive a vaccine. And some people seem to react when they have a vaccine, whether or not they've had COVID and whether or not they've had a first dose of the vaccine as well. So, and there doesn't seem to be any relation to getting a bad local side effects like a sore arm if you've got them on the first dose, and then you might have something different for the second dose. I think that's certainly true for the Oxford vaccine, which we've been studying. They just seem to happen or they don't. And the same's true if you've had COVID. Though anecdotally, you see a lot of people saying, oh, I had a bad reaction, I had COVID. So people are talking about that, but I don't think we've got the data to show that at this moment in time. And of course, about 10 to 15% of all the people who took part in all the big phase 3 trials had already had COVID. So we don't think that's true from the trials, although, you know, as you say, people are talking about that. So long COVID, I think, is increasingly interesting, isn't it? We've seen some reports in the newspapers over the last week or two where people have reported that the vaccine appears to be safe in the 40 or 50 people that they've studied with long COVID, but I think it's far too early to say whether the vaccine is going to help those people's symptoms. Lots of people in lots of different viruses and bacteria have a post-viral or post-infectious syndrome, and I don't think we have got evidence from any other condition that vaccines can make the symptoms from those post-viral and post-bacterial conditions better. So time will tell. It's certainly not going to do any harm. And I don't know whether Adam looks if he's turned his mic on. Did you want to add to that one, Adam, or is that
sorry, yeah, no, that was Sky News coming in. So I'm going to have to go, I'm afraid.
Okay, so this is a live webinar, you can tell. Thanks, Adam, for being able to stay with us for as long as you were able to. And thanks, everybody, for taking part in this evening's event. I think it was absolutely fascinating. I've been reassured about some things. I've had my confidence levels boosted. And thanks to you, audience, for asking such really, really good questions. I'm sorry we didn't have a chance to answer all of them. I still have quite a list, and that's both from the question and answer box as well as those that were submitted a little bit earlier. So I'm sorry we didn't have a chance to answer all of those, but you will receive a follow-up email after this event, and the NIHR will include answers to some of the questions that we weren't able to answer, but were asked by several different people, perhaps in slightly different ways. So that will be coming to you. Now, if you, like me, are interested in volunteering to support vaccine trials, again, you can sign up for information on the NHS COVID-19 Vaccine Research Registry. I'm going to give you the URL for that. That's nhs.uk/research contact. And Adam, were you going to tell me something? No, he's just waving goodbye. Oh, he's gone. Okay, so we've got 3 approved vaccines, and that's only been possible because of the numbers of people that have volunteered. So again, thank you very much for that. And do keep volunteering, because there's a lot more research that we can carry out gainfully for not just the UK, but to affect the rest of the world. So if you're interested in finding out more about other areas of research that you can volunteer to take part in, please visit, now this is a long URL, bepartofresearch.nihr.ac.uk. I'll read that one out again. bepartofresearch.nihr.ac.uk. So it just goes to me and the rest of our panelists to say thanks again for attending. And panelists, thank you so very much for sharing all your knowledge and your enthusiasm with us. We really, really appreciate it. Thank you so very, very much, and a very good night to all.